Well, thank you for the opportunity for the opportunity to share our work on the development of our environmentally friendly larvicide. So I'm Ivy Hurwitz. I work at the Center for Global Health in the Department of Internal Medicine. So let's just get started. Whoops. There we go. Oh, no, won't do it. Okay, there we go. Okay, so mosquito-borne diseases. They affect over 700 million people annually, resulting in over 1 million deaths. So the Aedes mosquito, or the tiger mosquito shown here, is the primary vector for viruses that causes Zika, dengue fever, yellow fever, and chikungunya. Malaria and West Nile diseases transmitted by other mosquito species. So except for yellow fever, there are currently no vaccines against mosquito-borne diseases. So besides protecting yourself from mosquito bites, vector control is perhaps the best solution to disease prevention. So to best understand how to best control mosquito population, we must first understand its life cycle. So, and that's shown here. So all mosquitoes require standing water to reproduce. So depending on the species, the female mosquito will lay her eggs either directly on the surface of the water or along the edges of the surface of the water just above the waterline. Once the eggs hatch, the larval stages begin. The larval of most mosquito species will hang out at on the water surface because it needs to breathe, and it does that through this little siphon that is attached to its tail. So as they feed, the larvae will molt, and it will undergo 4 molt stages as you go through the first larval stage, second, third, and fourth stage. And after the 4th molt, a pupa will emerge. Depending on the species of mosquito, the food availability, and the water temperature, the larval stage usually range between 4 and 14 days. When the pupa is fully developed, the skin will split and an adult mosquito will emerge. And then the female mosquito will go on to bite someone, it'll find a suitable mate, and the cycle begins over again. So if you look at this cycle, there are 2 obvious methods to control the mosquito population. One can use adulticides to target the adult mosquito that is flying around. So adulticides, which are usually either pyrethroid or permethrin-based solutions, are highly toxic, right? Not only to the mosquitoes, but also to non-target species. And because it's been used so much, there's been a lot of development of resistance. So this method of delivery is perhaps not the best, although mosquito control agencies will tell you that it is safe to be outside when they're fogging. Perhaps it's not the best idea. So the second method, or another method to control the mosquito population, is the use of larvicide. And this might be an easier approach to control these insects, as they're larvae, right? And they're contained within the body of water. They can't really fly away from you. So there are currently 2 types of larvicides. They're the chemical-based larvicides, and these are usually organophosphates. And as we all know, most organophosphates can be toxic to humans and also non-target species. They also pose a high risk to the environment. These things are also fairly expensive to use, requires fairly frequent application. And of course, there are development of resistance in many, many mosquito populations around the world. Another method of control of larvicidal control is to use of biological base methods. So the first one here is the use of insect growth regulators, specifically metroprene and pyroproxyphene. So these insect growth regulators, basically what they do is they interrupt the life cycle of the mosquito. It will not allow the insect to go past the pupation stage. These things are highly efficacious. However, they can also, again, be toxic to non-target species. The other biological base solution that is very commonly used is Bacillus thuringiensis. So the spores produced by this entopathogenic soil bacterium has been used to control pests since the 1920s. These BTI dunks are readily available at your hardware stores. While this method of control is very effective, there have been numerous reports that BTI is unstable when exposed to intense sunlight, and as such will again require frequent application. So the need for a cost-effective, non-toxic, environmentally friendly larvicide is urgent. So we know about essential oils, and what essential oils are is that they are volatile compounds that have a strong aromatic components that give a distinctive odor, flavor, or scent to an aromatic plant. These oils are generally non-toxic to humans in low concentration, and they're usually produced by plants for use in its protection against predatory insects. As such, many studies have demonstrated that essential oils have larvicidal activities. However, there are currently no commercially available essential oil products to date that can be used as a larvicide. And the primary reason for this is that because essential oils are rapidly degraded by sunlight. Further, although they smell really good, you really do not want to dispense a large amount of oils into the environment, as this can be detrimental not only to the environment, but also to non-target species. So basically what we need is a delivery system, and this is where yeast comes in. Whoop, sorry. Oh, there we go. So this is where yeast comes in. So it turns out yeast cells can be used as a biocompatible and biodegradable capsule for a variety of compounds. So loaded payloads are further protected against UV irradiation by the cell capsule. For storage, encapsulated yeast can be readily dried down and then rehydrated for use as needed. Finally, and critically for our work, it turns out mosquito larvae will preferentially consume and readily digest yeast cells. So that makes it easier for us. So how do we make a larvicide? It's a very simple task. We take fresh yeast, a selected essential oil, in this case, orange oil as seen here, and we agitate this and add some water to that. We agitate this at 40 degrees C overnight. And when that agitation is done, we wash the excess oil away from the yeast capsule, and then we freeze dry our sample. And it looks like this when it's done. And when we're ready to use it, we rehydrate a little bit of the larvicide, and then we add it to the body of water that we're trying to treat. So the setup for production is fairly minimal. We basically need a heated incubator for the shaking, for the agitation, a centrifuge for the washes, and a lyophilizer to dry the samples down. So cost for production is also fairly minimal. For example, the material cost for a single application of yeast encapsulated orange oil to a 5-liter container of water is only about 10 cents. So we have demonstrated that 2 of our encapsulated oils, specifically orange oil and fennel oil, are highly efficacious against the Aedes or tiger mosquito, both under laboratory and in semi-field studies. The plot shown here are dose response curves of Aedes aegypti larvae to either encapsulated fennel oil, shown in blue, or orange oil, shown in orange. The amount of encapsulated fennel oil necessary to kill 50% of Aedes aegypti larvae in 24 hours is under about 7.5 milligrams per liter. And for orange oil, it is approximately 20 milligrams per liter. When used directly, the amount of fennel oil or orange oil, if you added the oils directly to the water, the amount that you would need to kill a similar number of larvae is closer to 50 milligrams per liter. So we also have data showing that the encapsulated orange oil is active for at least 3 weeks following application. And when stored appropriately, the encapsulated larvicides are stable for at least 1 year. And we're continuing to monitor the shelf life of our products. So while our current, while our current production rate is at about 2 kilograms of larvicide per batch, we believe that our technology is readily scalable with minimal startup and material costs. The essential oils that we're working with are food grade and are currently recognized by the EPA as non-toxic. And we're further working with the EPA for approval of our technology. As mosquito-borne diseases, specifically those transmitted by the Aedes mosquito, are present all over the world. The market for our simple technology is basically to all mosquito control agencies globally. So part of our technology is already under patent, and we are very excited to meet with potential investors to further license our technology. So I want to conclude by thanking the various investigators who have worked on this project. At UNM, the optimization of encapsulation was performed initially by Dr. Michael Workman and is currently being led by Ms. Alexandra Yangling. Our colleagues at Fiocruz in Brazil, these are the people who are performing all our semi-field testing, and our colleagues at the Uniform Services are working on the mechanism of action of the ingested oils. And we're also working with Alex and Henry at the Lobo Rainforest Innovations, and they've been tremendously helpful in helping us get our technology to patent. Our work is currently being sponsored by the CDC, and with that, I will be happy to take any questions.